Hi everyone, welcome to Basic Science series. Today we will be discussing antibiotic resistance. So the topic, you can see all the different points that I'll be discussing in this video lecture, where we will be discussing different mechanisms of antibiotic resistance, how bacteria develop mechanisms against antibiotic. Right. First, we need to understand what are the antibiotics and how the resistance against antibiotic is being developed by the bacteria, especially. It's a very important topic when it comes to microbiology and the infectious diseases. And currently, what is happening is a lot of microbes they are developing resistance against leading antibiotics. So soon we are not going to have antibiotic available for those infections, and if that happens, it will be a challenging situation. It will be a very, very difficult situation for everyone. Right. So that is why this topic is really important. Now let's move on to the slide 1. Where I'm trying to basically mention few things. We'll start with what is antibiotic, right? So what is antibiotic resistance? First, we need to understand antibiotic. These are chemical compounds. These are molecules that can target bacteria, right? So I can write it over here, target bacteria. Now, antibiotic, the name is made up of anti and biotic, two words, right? One is against, biotic means living things. Here I'm specifically talking about bacteria. So that is why antibiotic are also known as antibacterial agents, and then another name is antimicrobial compounds. But I hope you understood the overall concept, right? So we are specifically talking about the bacteria here and how bacteria develops the resistance against these antibiotics or antibacterial agent. I think antibacterial resistance, that is why we use another term which will be relatively more specific to our topic. So antibacterial agents. What is antibiotic resistance? So what happens in that case is basically the organism, it develops different strategies. So suppose since we are talking about bacteria, right? So bacteria, this is a typical bacterium, gram-negative bacterium having flagella, right? This is an organism. It will have genetic material inside, other organelles, right? And based on that, further it can basically divide and make multiple different organisms and further grow in that manner. But what antimicrobial agents they do, they basically target multiple components. They can target DNA, they can target here the proteins, right? And then cell wall, I guess, right, of the bacteria. And because of that, bacteria is now no longer be able to reproduce, and because of that, it can decrease the growth of the bacteria. And why we need that? Because many of the bacteria, they are pathogenic in nature, and they can cause serious infections. So to reduce those incidents of infections, we need antibiotics, and that can basically decrease the bacterial population and then minimize the damage to the host, right? And antibiotic resistance, what happens is bacteria develops different strategies. Because of that, then what happens is antibiotic is no longer be able to target proteins, DNA also, as well as cell wall. There will be a lot of different types of changes. And then the targets that were there of the antibiotics, now the antibiotics will not be able to target them. And how it happens, that is the today's topic, right? Okay, let's move on to the main points about the antibiotic resistance. That few of them we have already covered. First is it's a natural evolutionary process. Now, understand this. When you're targeting one organism that wants to survive, it will develop or it will evolve various mechanisms naturally, right, to basically block the action of that toxic agent. Because of that, it's a natural evolutionary process. Second, bacteria develop different mechanisms. So it's not only one mechanism, you have multiple different multiple mechanisms. So I'll write it over here. Multiple mechanisms through which bacteria can basically target these antibiotics and then reduce the effect of these antibiotics. So antibiotics are no longer effective. Now, challenge of antibiotic resistant infection. That is what I was talking about. Nowadays, what is happening is the infection caused by various organisms like Staphylococcus aureus, pathogenic E. coli, Pseudomonas aeruginosa, Acinetobacter baumannii, Salmonella, Shigella. There are so many organisms that are able to cause opportunistic infections in humans. They are developing antibiotic resistant strains or antibacterial agent resistant strains. Because of that, antibiotics are no longer effective. So now we don't have any option available right? And just imagine the chronic infections due to these antibiotic resistance bacteria can basically cause a lot of damage in the host, and the chronic infection will be very difficult to treat if we don't have antibiotics against them. So it's very, very important to understand antibiotic resistance and their mechanisms and develop alternative antibiotics or antimicrobial agents that can target those antibiotic resistance strains. Now let's understand a few of the mechanisms that I've I'm trying to explain those over here. Let's move on to the first one, which is mutations. Now we all understand what is mutations, right? So this is the DNA, and DNA is the molecule that contains the hereditary information. In DNA, you have the molecules A, T, G, C. I'm just writing the sequence, typical sequence of DNA. It's a repetitive sequence of these nitrogenous bases. I hope that is clear. Now what happens is sometimes what happens is bacteria develop some kind of mechanism. Because of that, there is a change in the sequence of DNA. There is a change in sequence of DNA, and you have another, suppose just instead of T you have A in the sequence. Now this is the mutation, the change in the base in the sequence of DNA, and that will basically change the target site, because this DNA is going to produce the protein molecule, and protein molecule is nothing, it's a globular molecule made up of amino acids, and we know that that triplet code basically gives one amino acid, right? So you will have amino acid 1 over here, amino acid 2 over here, and because of this change you will have alternative amino acids. The important point is when you have a significant change in this sequence, you will have a new amino acid. So new amino acid will be there, and that will cause the change in this protein, and that change is going to change the target site of antibiotics, obviously, if that happens in that particular gene, which is the gene for the target site of the antibiotic, right? And then these spontaneous mutations, they are happening, and that is very important for evolution process that is constantly happening. Because of that, they are rapidly changing the target sites, and the target sites, because the target sites, they are getting changed. Now antibiotic is no longer be effective. Another point is binding effectively to their intended targets. Now this can also change the binding efficacy of the antibiotics. So just imagine if this is the antibiotic. Now without mutation, it is able to bind correctly, and with mutation, it is not able to bind effectively to the target. So binding effectively to the intended target is reduced. That can also lead to the less activity of the antibiotics. Now because of that, the antibiotic will be less effective, or it can also become entirely useless if the mutation is significant. If the spontaneous mutations are significant, then it can also become entirely useless. Example is sometimes there is ribosomal DNA mutation, and that can basically, which is known to inhibit or reduce the activity of tetracycline antibiotic, and this is one of the protein. Its target is protein, right? So if there is a change in this one, right, that can affect because this is also the target site of this one, and then this can reduce the activity of the antibiotic. So activity will be reduced. Correct? Okay, so this is important point. Now let's move on to the second slide. I hope this is clear. The point 1 is the mutation. Because of the mutation, antibiotics are no longer able to bind to their target sites. Next is enzymatic degradation or inactivation. Enzymes again are the proteins that bacteria can produce, right? So enzymes, they are the protein molecules, and the role of enzyme is to basically target a substrate and change it into product. It's a general definition of the enzyme. So enzyme, they are protein molecule. They can basically chemically modify or destroy antibiotic. They can do both of these functions. Enzymes, they can modify chemically the antibiotic. Now antibiotic is no longer able to bind to the target. Second thing, enzymes, there are various enzymes who can do that. There are various enzymes that can directly bind to the antibiotic. They can degrade the antibiotic. So you can see over here, this is the penicillin antibiotic, and this is the beta-lactam ring over here. This is the beta-lactam ring. That is why penicillin is also known as beta-lactam antibiotic, right? So this is the beta-lactam ring. Now you have beta-lactamases, right? So for penicillin, beta-lactam ring is there. Now you have beta-lactamases. They can basically target this beta-lactam ring and then break it up. Now because of that hydrolysis reaction, because they can hydrolyze beta-lactam ring, the penicillin is no longer effective against the bacteria. Bacteria are they are developing resistance by the production of beta-lactamases, and there are different versions of these beta-lactamases. They are making the infections really, really difficult, and a lot of research is going into this direction, that how beta-lactamases they are degrading antibiotics and how we can produce inhibitors. And you will see nowadays antibiotics are coming with beta-lactamase inhibitors in combination with beta-lactam antibiotics. That is another strategy, and people are prescribing those kind of doctors, they are prescribing those kind of molecules because it makes sense, right? You can have antibiotic resistant infection without knowing it. You can directly use beta-lactamase inhibitors. So it's a very, very attractive area of research over here, where you can work on identifying novel beta-lactamase inhibitors, right? So that is point number 2, enzymatic inactivation. Efflux pumps. So this is one schematic that I've designed. I will show you actual structure of efflux pumps. The efflux pump that I've given the example over here, ACR-AB-Tol-C, one of the efflux pump that is known to be present in various organisms. Now what happens in this case is basically this is present on the membrane. So just imagine this is the membrane. Now antibiotic is somehow able to enter inside the cell. Efflux pump, they will have the ability to basically take the antibiotic and basically throw the antibiotics out. They will just take the antibiotics and throw it out, right? That is why they are also known as pumps, efflux pumps, right? And then there are multidrug efflux pumps. The example I've already told you, ACR-AB-Tol-C system, which will effectively pump out the antibiotics from the bacterial cell, and they are specialized protein. They are not single protein. So you can see you have domain 1, domain 2, domain 3, right? Different domains are there, and because of that, obviously you need different domains, because it will have one part that is embedded in the cell membrane, another part is exposed to the outside, another part is exposed to inside. So different types of domains are there, although this is the schematic, right? It's not the actual structure of beta-lactamase, not beta-lactamases, right? We are talking about efflux pumps. This is not the exact structure, it's a schematic, but as you can see over here, it can really be helpful to explain you if you have multiple domains like this. I'll show you the structure of actually efflux pump after this, but I hope this is clear that they are specialized protein. Means they are not very simple protein, they are complex proteins. They have special function. They will pump out the antibiotics. They will reduce the intercellular concentration of antibiotic by just pumping out the antibiotics. So you can see over here, if the antibiotic is there in the system, they can effectively pump them out. These are the examples. Now let's move on to the actual structure. Look at this. This is the PDB, protein data bank, PDB file or structure. This is the crystal structure, a cartoon representation, I would say, of this efflux pump. So you can see over here, some things, they are in green color, some are in purple. They are different domains, different, and then you have this repetitive kind of helical structure, and you need, these alpha helices. They basically they are important when it comes to membrane proteins. So this part is going to be present in the membrane. Then you can see it has a kind of a channel system in the center. It's a very interesting molecule. It's a huge molecule. So it can basically do this, right? The expel out the antibiotics. It can pump out the antibiotics and then reduce the concentration of the antibiotics. So this is how it actually look like. It's not the exact structure. It's a cartoon representation where you're showing various amino acids, but actually you can see how the molecule looks like, right? Now, next mechanism, cell permeability. Cell permeability means bacteria, suppose this is the cell wall, bacteria can have this mechanism also, where bacteria will change its permeability, and now antibiotic first is no longer be able to enter inside it can basically do that, and now antibiotics are no longer to be able to enter inside the bacterial cell, right? So I think I also need to make a DNA, right? Genetic material is the part of the molecule, a part of the molecular system, part of the bacteria, right? So I'm creating this illustration. It looks good, correct? So antibiotics, they cannot penetrate inside the bacterial cell. And how it can do that? It can modify the lipid environment of the cell wall, right? And we know that the structure of the bacterial cell wall contains lipid, peptidoglycan, all these molecules, they can be modified by bacteria. Because of that, it is very, very difficult for the antibiotics to penetrate inside the bacteria. And Because of that, it is not able to reach to the target, and this mechanism is known basically in gram-negative bacteria. Gram-negative bacteria is known to change the cell wall of the bacteria by this mechanism, by modifying the permeability. How it, what is the mechanism? It alters the outer membrane. That is the first target, because outer membrane is the membrane which is exposed to the environment. If there is change in that membrane, what will happen? Antibiotic will not be entering into the system, limiting the entry of the antibiotics, right? And there are various antibiotics that they are blocked in that way, and various gram-negative bacteria, they have the ability to modify their outer membrane. Horizontal gene transfer. This is another mechanism. It's a very important mechanism. So you can see over here, bacteria 1 and what says bacteria 2. What is happening? This bacteria, it has developed the resistance, and it has extracellular or extra chromosomal DNA, which is known as plasmid. This plasmid will have antibiotic resistance gene. It becomes arginine. It's not arginine, right? ARG. So I should be avoiding that. So antibiotic resistance gene. It will have that antibiotic resistance gene over here, and that gene will be directly transferred to another strain by horizontal gene transfer. And for that, you have 3 mechanisms. Mechanism number 1, conjugation. Second one, transformation, transduction. I'll be making separate videos on all these topics, so please stay tuned and watch all the videos that I will be posting. I'll explain all these points in very much detail. Right now, I'll just tell you that you have 3 different mechanisms by which bacteria can transfer its genetic information to another bacteria, and it's also known as the horizontal gene transfer of the information from one bacteria to another one, right? So what is happening? Genetic material is getting transfer horizontally to the same population, right? Not to the next population. So it's horizontal gene transfer, and it can cause the rapid spread of antibiotic resistance gene. I'm sure you have seen one cartoon is there where one bacteria is giving the antibiotic resistant gene to the another bacteria. You will see, just type antibiotic resistance and go to the images. You will find out that one bacteria is giving the bacterial gene, and the gene that is basically is being given through the exchange of the plasmid, and plasmids are the extracellular, extra chromosomal genetic material. Okay. Now it can cause rapid spread, and how it can happen? It can happen through the plasmids, right? So I hope now that is clear, that we can have this information transferred by using horizontal gene transfer. Moving on to the next one, biofilm formation. This is one of my research area, one of the area that I'm very, very interested in, biofilm formation. And our lab is working to understand how these biofilms, they are getting produced, and what is the mechanism of the biofilm formation. Biofilm is not just the clumps of cells. It is a complex community of microbes which contains a lot of material, like it will contain extracellular matrix, which will composed of polysaccharides, proteins, DNA even, and then other small molecules, signaling molecules will be there. Quorum sensing will be happening in those communities. It will be protected by the matrix, right? Matrix will be there. Cells will be different in a biofilm mode than the planktonic mode. It's very, very interesting and dynamic process. And then the entire biofilm can function as a multicellular organism. That is another important aspect, that where all the cells, they can be synchronized and they can be behaving like one single organism. Anyways, watch my video on biofilm, how biofilm gets formed. I made multiple videos on this topic. Please watch those videos. Here, biofilm formation we'll discuss in the context of antibiotic resistance. Right now, what are biofilms? They are communities of microorganisms that I've already explained, encapsulated or encased in the matrix. They can also help in resistance to the antibiotics. How? Because they are encapsulated, or they are protected by the matrix. Now antibiotic is no longer be able to enter into the biofilm, and then you need very high concentration of the antibiotic. Now because of that, bacteria will develop resistant phenotypes against that particular antibiotic, and that is not good. It protects the bacteria, obviously, as I told you, that when cells are all together, unity is the strength, right? You know that. So this is the same concept over here. Example is chronic. In case of chronic infection, what happens is bacteria slowly survives. It develops resistant phenotypes, and that forms the biofilm, and it can cause chronic infections. Chronic infections in various organs. They are known to be present in various organs, and especially when there is an implant, and then the patient can suffer from chronic infections, especially with the biofilm formation. Correct. since they are very difficult to treat, biofilm can lead to chronic infections. So that is why they are also highly challenging to treat. We need very high concentration of antibiotics. There is a research on this topic that we need to find out alternative strategies because we don't have too many antibiotics, right? And people are also not working on developing antibiotic. One reason is that they are not that expensive, but antibiotics should be expensive. Only that will force everyone to reduce its use, and also a lot of researchers, they will work on this one, because money is something that I don't, for some reason which is not right, controls the direction of research in many aspects. Anyways, that is the biofilm formation. Next is significance. Now, why the antibiotic resistance is so important? One, mechanism. Understanding mechanism is very, very important, because if we understand the mechanism, then only we can develop therapeutics. So to develop therapeutic strategies to combat antibiotic resistance, is very, very important to understand the mechanism, right? I've already told you that the horizontal gene transfer is another mechanism. So we can find out strategies that will reduce that horizontal gene transfer. We can find out novel beta-lactamase inhibitors. That is another strategy. Responsible use of antibiotics. Now, every nation is very, very serious about the antibiotics, right? So there should be guidelines, there should be policies that will be controlling the use of antibiotics, and it should be used responsibly. And awareness regarding antibiotics is very, very important. There should be programs, community-wide programs, where people can introduce the uses, how the antibiotics should be used in communities. They should be used when they are prescribed, not like that. And the antibiotics should be, the use should be monitored by the trained physician. All those things should be there. They should not be available without prescription, and all those things should be there, right? And development of new drugs, that is something which is highly important. And I don't know why, people, especially those policymakers, they should force this policy that new drugs should be developed against antibiotic resistance. The projects should get funding which are related to this topic. Ongoing research is very, very important to stay ahead of the war that is going on between bacteria and antibiotic. And obviously, let me tell you one thing: bacteria is on the winning side, and it will be on the winning side, because antibiotics are basically not they are not evolutionary product of the evolutionary process, right? We are the one, humans. So it's a kind of let me also explain again. Humans are producing the antibiotics to control the bacteria. Now, bacteria developing resistance naturally, and that antibiotic is basically not getting developed naturally, because human effort is going into that direction, and that is taking time. Here on the other side, bacteria is basically it's the organism which grows very, very fast, and evolution in bacteria is going to be very quick, and they are here long before humans were. That means they are able to adapt to any natural condition. So what we need different strategies. We can have different organisms, especially bacteriophages, and a lot of people are working in that area. And one of the strategies that we are also working in our lab is developing phages. When you develop phage, what happens is you develop a biological system, although they are not entirely living. But they can also adapt to bacteria. So it will be a constant war between bacteria and bacteriophages, not bacteria, antibiotic, and humans, right? So we need to have that particular strategy in place. If the research is going into that direction, and we are developing different types of phages that can resist the immune system, because the problem with the phages is our immune system clears the phages very, very quickly. So that is another. A lot of challenges are there, but I think the other countries like Georgia and Russia, they are working on phages, and they have developed a lot of advanced technologies when it comes to phages, and we are not doing that. We should be doing that. We should not be following the route of antibiotic. We should be following the route of bacteriophages also, and then using phages to take care of these infections. And this is very, very interesting to me. So we are working in this area, developing new strategies, developing the phytocompounds, developing non-antibiotic compounds that can reduce the bacteria, developing antivirulent compounds that can reduce the virulence of the bacteria, and also antibiofilm compounds and phages, right? So this is very, very interesting topic. This I hope I was able to give you an overview of this topic. If you want further information, you have these references. Although there are so many papers and review articles, books you can read on this topic, these are just few references that you can read. I've used them to prepare this lecture, so you can read these, and they are going to give you a lot of more detailed information that is beyond the scope of this lecture, right? I can't be making a long video, and it's very difficult for me to make a 2-hour long video. So, and these are very big articles, right? So one article that is there, antibiotic resistance in the environment, published in Nature Review Microbiology. So that is very important article, and I recommend you read this article in detail if you are interested in antibiotic resistance. Second, molecular mechanism of antibiotic resistance revisited, another very important article, and then you have antibiotic resistant bacteria, current situation and treatment options to accelerate the development of new antimicrobial or antimicrobial arsenal. Right, this is expert review, anti-infection therapy. Anyways, so these are some of the articles that I recommend, and you can obviously go to the reference section of these articles and read those cited references. Correct, and because of that, what you will get lot of good articles in the area of antibiotic resistance, and that will give you initial knowledge pool which you can utilize to study more in the area of antibiotic resistance, right? So I think this is the end. Yeah, this is the end of the topic. Okay, so this was all that I wanted to include in this topic. What we have covered so far is what is antibiotic resistance, why it is important, what are the different mechanisms against antibiotic and bacteria, how the bacteria is developing resistance. All those points we have covered. I hope this video is helpful for you to understand the antibiotic resistance. I will meet in the next video where I'll be bringing more interesting lectures and topics like this. Thank you for watching the entire video, and I know it's a lot of information, packed information, not that entertaining. And when we use mobile phones and computers, we used to see a lot of entertaining stuff, but this is something that that is I create. If you like the video, then please do hit the like button, and then if you want to see more videos like that, then please do subscribe to the channel. Okay, I'll meet you in the next one. Till then, take care.